welcome you to lesson 4, and today who can guess what we are going to look at? Yes, we are going to look at enzymes. Wow, you must be very lucky to attend to this lesson because we are going to look at something very important in the topic of nutrition, and this one is what we call the enzymes. So what are the enzymes now? I know you must have heard about them. You probably have read about them from some of the textbooks. Well, so we want to define an enzyme together. So let's define what enzymes are. So enzymes are organic compounds that are protein in nature that speed up the rate of biochemical reactions in the body of an organism and remain unchanged at the end of reaction. Wow. These are great things, I see. So can we refine it again together? Enzymes are organic compounds that are protein in nature. They speed up the rate of biochemical reactions in the body of an organism and remain unchanged at the end of reaction. So enzymes, for now at least, we know that their work is to speed up the rate of biochemical reactions in the body. Sometimes we say they are metabolic reactions. Instead of saying biochemical, you can call them metabolic reactions. All those reactions that take place in your body. I know you know some of them: respiration, digestion. Which other one do you know? So all those reactions that occur inside our bodies are called biochemical reactions. So enzymes help or facilitate these reactions by speeding them up. So that means enzymes are important. That basically means enzymes are important in our bodies. Imagine that we don't have enzymes in the bodies. How would business be in our bodies? Will it be business as usual? Probably. So the importance of enzymes include the following. I want us to know that the rate at which some of the reactions occur in the body without enzymes would be too slow to sustain life. If we did not have enzymes in our bodies, some of the reactions in our bodies would be so slow. Yes, imagine that you have eaten food today and then digestion is completed next month. Man, how would you get energy? How would you repair and build tissues in a body? It would be so slow, and life would not be able to be sustained. So therefore enzymes are vital. Enzymes are therefore important because they speed up the rate of reactions without changing the product formed and the nature of the reaction. So enzymes do not change the product formed. For them, they just speed up the reaction. They cannot make the reaction that would not occur to take place. They cannot initiate any reaction to start. And they cannot make an endothermic reaction to become exothermic. They do not have the ability to change the nature of the reaction. But what do? They just ensure that the products formed are formed in the shortest possible time. Therefore, they speed up the rate of what? Reaction. If a reaction was supposed to take 3 minutes without an enzyme, if you introduced an enzyme into that reaction, it would take 1 minute or 2 or less. So that is the role an enzyme does. So they control metabolic processes, hence they promote the normal body functions. Yes, they promote normal body what? Functions in the body. So let's look at, having known that enzymes are now important in our bodies, let's look at now the classifications of enzymes. Enzymes are classified basically basing on the nature of the reaction that they catalyze or the nature of the substrates that they act on. So enzymes can be classified based on that, based on the nature or the type of reaction that they catalyze. For example, let's look at the following classes of enzymes. The first one, there are enzymes that are called the isomerases. Yes, I know you know the word isomerism. Isomers. You remember this in your senior 2 chemistry, in your organic chemistry. Yes, in senior 2 and senior 3. Isomerism. So we also have enzymes that are called isomerases. So these are enzymes that catalyze reactions that involve isomerism. For example, you know some of the molecules in our bodies that undergo isomerism. One of them is glucose. Glucose exists as 2 isomers. Actually, more than 2. The common ones are alpha glucose and beta glucose. So the reaction that lead towards formation of such isomers are catalyzed by a group of enzymes called isomerases. Wow. Then we also have phosphorylases. These are enzymes that catalyze reactions involving addition of phosphate groups in 2 molecules. For example, the reaction that leads into formation of ATP, adenosine triphosphate. Adenosine triphosphate is formed from adenosine diphosphate by addition of a phosphate group into adenosine diphosphate to form adenosine triphosphate, ATP. So that reaction is catalyzed by phosphorylases. Yeah, it's catalyzed by phosphorylases enzymes. Then we have hydrogenases. These catalyze reactions involving addition of hydrogen into molecules. Then we have dehydrogenases. They catalyze reactions involving removal of hydrogen from the substances. Yes. Then we have kinases. These catalyze reactions involving movement of molecules from one area to another. From the word kinesis or kinetic energy, movement. Yeah, they catalyze reactions involving movement or transfer of molecules. Then we have carboxylases. These catalyze reactions involving addition of carbon dioxide into substance or into solutions. We also have decarboxylases. This involve reactions involving removal of carbon dioxide from solutions or substances. Well, so we have those classes of enzymes. I want you to master them. Master the class of enzymes and also master the reaction that it catalyzes. That will be very good for you, and summarize it in your book. Isomerases, phosphorylases, hydrogenases, dehydrogenases, kinases, carboxylases, decarboxylases. That will be great for you. So members, enzymes can also be described as intracellular enzymes or extracellular enzymes. So what are those ones now? Intracellular enzymes are those which catalyze reactions inside the cells that are producing them, e.g. the respiratory enzymes. Those ones are intracellular. Yes, enzymes are intracellular. Those that act, that are responsible for conducting processes like glycolysis, Krebs cycle, and so on, as we shall see later on. Those enzymes there that are involved in the production of energy, the breakdown of glucose to produce energy, those ones act within the cells that produce them. They are called intracellular enzymes. Then we have extracellular enzymes. These are produced by one cell, but they catalyze reactions outside that cell that produces them. For example, all the digestive enzymes are extracellular, though you know some of the enzymes that are involved in digestion. For example, pepsin in the stomach, salivary amylase, trypsin in the duodenum, renin in the stomach, and so on. Those enzymes there are extracellular enzymes. There are cells that produce them, but after producing them, they are secreted into surfaces which are far away from those cells that produce them. So we can categorize enzymes based on which part do they act on. Is it within the cells that produce them or outside the cells that produce them? They can either be intracellular or extracellular. I want you to add that also in your summary. What are intracellular enzymes? And what are extracellular enzymes? That would be great. So we are going to move to something else called nomenclature of enzymes. Nomenclature means naming, giving names. So how do we name enzymes? So enzymes are named by adding the suffix "s" to these substrates or to the substrates that they act on. S, a suffix is an ending word or a group of letters that are found at the end of the word is called a suffix. So a substrate is a substance which the enzyme acts upon, or simply a raw material for an enzyme. So when you know the name of the substrate, at the end you replace the last letters with the word "s", then you have the name of the enzyme. For example, we have some of the enzymes here and their substrates. We have a substrate peptides, and the enzyme is peptidase. Yes, peptides are protein molecules. Then we have substrate lipids, and the enzyme is lipase. Then we have maltose. The enzyme is maltase. We have sucrose. The enzyme is sucrase. We have substrate lactose, and the enzyme is lactase. We have a substrate cellulose. The substrate is cellulase. So you have seen how interesting it is. You just replace the suffix of the substrate with the word "s". Maltose, maltase. Sucrose, sucrase. Lactose, lactase. Cellulose, cellulase. And so on and so forth. That is how we carry out nomenclature of enzymes. If you know the substrate, you can easily name the enzyme as well by adding the suffix "s". Then we have some enzymes which retained their names, the names they had before. Before the nomenclature was introduced, all enzymes had their original names. So, but some of the enzymes, even after the nomenclature was introduced, they retained their original names. For example, enzymes like pepsin and trypsin, those ones retain their names. So they don't conform to the current nomenclature of the suffix "s". They just maintain their names. Pepsin is found, of course, in the stomach and acts on proteins. Then trypsin is found in the duodenum. Then we have, of course, other enzymes as well of the kind. So we must be cautious to know that most of the enzymes conform to the nomenclature of suffix "s", but also there are some of the enzymes that maintain their original names. Yes, like pepsin and trypsin. And then sometimes the enzymes digesting carbohydrates are generally called carbohydrases, and those that digest proteins are called proteases. This is a general name. For example, when you look at sucrase, maltase, cellulase, galactase, those are enzymes that digest carbohydrates, because maltose, galactose, and so on are carbohydrates that we have mentioned. So those enzymes have a general name. We can give them a general name, carbohydrases, because they act on carbohydrates. Then we have enzymes like protein, like pepsin and so on. Those ones act on proteins. So we have a general name, proteases. Proteases meaning they act on what? Proteins. So you can also group them like that. So members, nomenclature of enzymes has been interesting, hasn't it? Yes, and I know you have enjoyed it. So we want to move to something called properties of enzymes, and this one you are going to enjoy it more. properties of enzymes. So we must be interested to know, as scientists, yes, about the properties of enzymes. Yeah. So let's look at them. They are quite a number, but we want to look at them, and I will be briefly describing and explaining them as we proceed through very fast. One of them is they are all protein in nature, meaning enzymes are made from proteins. So therefore they are protein in nature. Or better enough, we can say they are actually globular proteins. Secondly, they are specific in action. So they catalyze specific foods only. For example, maltase only acts on maltose. If you brought sucrose, maltase cannot act on it. So they are very specific, only to a given food substrate. The other one is they speed up the rate of chemical reactions, meaning they are catalysts. Or better enough, we can say they are biological catalysts because they speed up rate of biological or biochemical reactions in our bodies. The other one is they are effective even in small amounts. You don't need to have a lot of enzymes for reactions to occur. No, even in small amounts. After all, they can be reused time and again and again. So even a little amount of enzyme can act